Hello, everyone, and welcome to Cell and Gene Live. Our topic is scaling cell therapies, auto versus allo, and I'm your host, Erin Harris. I'm joined by two of the best and brightest the sector has to offer to discuss our topic. Michael Blackton, SVP Cell Therapy Manufacturing at Nurix Therapeutics, and John Lee, VP Head of Cell Therapy at Center for Breakthrough Medicines. We're going to dive right into our discussion in just a second, but before we do, please note we will be taking audience questions. So submit your questions for Mike and John via the Q&A tool, and we will get to as many as we can. Also, I'll be asking you to respond to 2 poll questions throughout the event. So please take a moment to respond when prompted. All right, on to the good stuff. So if you're a repeat viewer of Cell and Gene Live events, you know I like to start out by setting the stage and level set the conversation around our topic. And so scale up of a process involves using larger vessels to increase the volume of production. In scale out, the culture vessel remains the same, but more units are added to a system to increase production capacity. And any discussion regarding cell and gene therapies inevitably includes the challenges of manufacturing them at scale. And so, John, I'd like to start with you. Broadly speaking, talk to us about where we are as a sector with autologous scale out.
Yeah, it's we're at a critical, almost an inflection point right now with regards to scaling out autologous therapies. We have almost a handful of FDA approved cell therapies, in the oncology space, as well as some others in non-oncology applications. And, we have another 1,500 or thereabouts clinical trials underway. So understanding what's going on with scaling out and the limitations of potential solutions is extremely important. Kite kind of leading the way there. Early on, Kite obviously had their manufacturing in California. They expanded into Europe, into the Netherlands, and more recently opened up another facility in Maryland. J&J obviously is doing similar things now as they ramp up to increase commercial production for Carvykti, their BCMA asset. As I see it, there are a couple primary challenges. One is around regulatory. The regulatory environment, obviously the FDA and European Medicine EMA they require as innovator companies and manufacturers begin to branch out and create their products in other venues, there needs to be a comparability assessment that's done, and this is a key thing that needs to occur as we're scaling out for those autologous products. Similarly, just from an operational perspective, unlike allo, where scaling up is necessary, the scaling out really it falls on a number of things. Obviously, you've got the facility, you have the equipment within the facility, as well as the personnel. And as a general rule, the world doesn't have enough qualified personnel at the moment to really scale out as effectively and as quickly as we would otherwise want. In terms of solutions, I think there's a couple ways that this can happen in autologous therapies. Reduced harvest manufacturing is an area that is gaining steam. Novartis last December, at ASH, they released or described their quote T-Charge platform. This is a manufacturing platform that allows for harvesting of autologous cell products in about 48 hours. And Gracell is another company that's also doing this. And what this enables then is really to maximize efficiency within a manufacturing facility. When vein-to-vein times were previously anywhere from 14 to 28 days, some of these new manufacturing, reduced harvest manufacturing processes are really bringing that in to just a few days, which allows then to really increase the efficiency of those same facilities. And then, of course, it goes without saying, but I'll say it anyhow, obviously integration of digital capabilities, manufacturing execution systems, electronic batch recording, automation, and so forth are other potential solutions, but these are more in their nascency at this point in time.
Yeah, good point. You bring up a lot of topics, even as you just talked there, that we're going to cover throughout the rest of our hour. But before we do, I want to make sure that we hear Mike's perspective on this. Mike, from your perspective, thoughts on what John just said, in addition to your take on the sector as from an autologous scale-up perspective, where are we?
I think from an autologous scale-up perspective, we're really looking at two things. One is, scale up or scale out within the facility, and that's where you get into cross-contamination issues and ensuring product safety within that facility. And there's various facility designs. There's, we probably heard the term ballroom concept, which presents some opportunities and challenges versus individual rooms per patient, for example. The other aspect is scaling out across geographies, because you're dealing with not only supply chain issues across different geographies, but also jurisdictional issues with different regulatory requirements. But I think it all comes down to life cycle practices. If you have adequately thought about your process and developed that process with consistency in mind, taking into account aseptic processing requirements, for example, cross-contamination, patient scheduling, then you're well suited to a scale-out scenario. Now, rather than talk about individual technologies, I tend to think of these in terms of broad brushstrokes, because every product is different. But remember, if you have an engineered autologous product versus, say, a TIL, there are different requirements. For an engineered product, we have vector control and supply chains versus the cell therapy control and supply chains. You really have kind of two products there. And how does that relate to your scale out scenario? So if you have one source of vector, for example, and multiple geographies where you have cell therapy processing, that presents a different challenge than if you had multiple starting material or vector manufacturing sites and multiple geographies where you're scaling out. So that's kind of the way I look at it. And the other thing I think is important with these, and I'm a real advocate of automation of these processes. Again, requires a lot of process capability and control. We have to think about the core requirement, and those core requirements are life cycle, post-approval changes, right? So we should always assume that we want to develop our product for future changes to make that happen in a expeditious way. And John talking about comparability is a good example of that. What are we comparing? How are we comparing it? What constitutes comparability? It doesn't have to be exactly the same, or is it substantially similar? What do these terms mean? It's all different for every product.
Sure, that makes sense. I want to have the exact same conversation, but from an allo perspective. We have FDA approved autologous therapies, but where does the potential lie for allogeneic? John, let me ask you that. Let's just have the opposite conversation.
Yeah, with allo, I think it's a bit earlier. We're still really trying to focus in on some of the rate limiters, which are largely include downstream processing, harvesting, formulation, those kinds of things. We're dealing with increased volume there from a scale up perspective. But there's an underlying issue with allogeneic, and that is really maintenance of the integrity of the drug product, as measured by functionality. You can imagine, there's a big push, especially in the world of T cell immunotherapies, to adhere to this stem cell memory-like phenotype. And as you try to achieve the numbers of cells that we're talking about in the allogeneic world, a typical harvest, at least the goal is somewhere between 10 to the 11th and 10 to the 14th cells per batch. To achieve that level of expansion, the possibility exists that you're going to end up with a phenotype within that population that are more differentiated than ones that have not been expanded to such a degree. So finding that balance between expansion as well as functionality is a key component here. And then, downstream from that, those more logistical or operational constraints that I mentioned with regards to downstream processing, in some cases, 2,000 liters worth of material to process, filling that drug product on the back end and maintaining consistency from vial 1 to vial 500 is it's a logistical constraint that folks are contending with at the moment. But I think, as we're discussing, it's a problem that is not completely solved. And, the field is getting there. We're seeing fillers with better capacity and mixers that will accommodate microcarrier-based cell culture. But at present, a one-size-fits-all solution to some of the problems that are facing the allo scale-up are just not available as of today.
Yeah, you're not kidding. We talk about that on Cell and Gene all the time, how there's just several problems that have yet to even come up and to be figured out. Mike, I want to get your weigh in on this. Before we do, I'd like to put up our first poll question, which we'll get to later in the hour. But Mike, what's your take on the allo scale up? Where are we, and where does the potential lie?
So, my background is in monoclonal antibodies, and monoclonal antibodies back in 1995, 94, we thought these were not characterizable. We're never going to be able to make these large volumes. I mean, we were in very small bioreactors, hollow fiber systems, for example, adherent cultures. But today, monoclonals, it's not even CBER, it's CEDAR. It's very rote. I see that with allogeneic. The problem is, I think the technology is very much earlier than it was even in 1994 for monoclonals, where you have for autologous, you have scale out. For allogeneic, of course, we have this opportunity for scale up. But what does that really mean? There are other issues, right? We have for autologous, we have starting material variability. Each patient has different starting materials, so you have to address that. With allogeneic, we have cell banks to deal with, and there's some allogeneic products that have multiple cell banks that have to be managed and characterized. Right, that's very similar to the starting material challenges we have with autologous, but the benefit is you have a consistent amount of starting material and your cell banks are consistent. The other piece that you really don't have with autologous is this donor eligibility piece, which I know is not really germane directly to manufacturing, but that is a concern. And the genetic manipulations we do as we develop these allogeneic products could become a challenge because, of course, we don't know what all the oncogenes are. One of the concerns with developing stem cell allogeneic products, of course, is oncogenesis. And then the other piece I have on my notes here is, long process, right? So some allogeneic products, while you can scale up to, 2,000 liters or more, some of these processes can be months long and have multiple genetic manipulations and scale up issues kind of serially. But once you get that solved, it's very conducive to scale up. Long process. I'm worried about aseptic fitness, right? Like container closure integrity and process integrity. While we are more, conducive to traditional scale-up pathways, it really remains to be seen whether you could go beyond, 3 to 5 liters in a cell culture system. The other thing that becomes important with allogeneic is this notion of real-time process monitoring, which we've been struggling with in traditional cell culture for years. I think it becomes very important given the length of these processes and the aseptic issues. So if we can have, online cell counts and of course pH and all the other parameters and be able to react to those and characterize using those. That would be really important.